So our third learning unit is on protein structure. So it's actually 3 chapters from your book. A lot of the material that is in chapters 4 and 5 are either things that we sort of discussed outside of class, or again, were part of your fundamentals quiz, which was amino acid structure, or are concepts we're going to discuss in lab. So again, some of these early slides on chapters 4 and 5 are going to be fairly brief. So we did talk about structure of amino acids, but mainly, again, you guys learning and taking your fundamentals quiz on amino acid structure. We talked briefly about some of the chemistry that can happen between amino acids. And so this slide allowed us to think about the chemistry that happens between 2 functional groups that all amino acids have. They've got a carboxyl group and they've got an amine group. Again, each amino acid having an N and a C portion. So we talked about condensation chemistry to kind of generate this peptide bond. Reaction mechanism 3 is where we kind of see the biological equivalent of that, where we have chemically or biochemically activated carboxylate groups with a good leaving group able to react enzymatically with what equates to a poor nucleophile in our protonated amine there. But again, that chemistry can happen in the context of an enzyme active site, not chemistry we're really going to study in this class. But condensation chemistry, by eliminating a small molecule like water, allows us to generate a peptide bond. So one of the skills that you are going to have to know for your exam is how to draw a small peptide. So not only being able to draw the peptide backbone, remember we've got N-C-alpha carbonyl, N-C-alpha carbonyl, but being able to draw the relevant R groups depending on what I tell you the amino acid sequence is going to be. And that is going to be something, it literally is going to be a word that I give you. And so you'll need to take the letters of that word and be able to draw the amino acids. Beyond that, it's also being able to identify different parts of an amino or a little peptide structure, such as identifying the amino terminus, the carboxy terminus, and then the direction that we have this peptide kind of proceeding in, always drawn N to C. Identifying the appropriate linking bond, and in case of peptides, amino acids are linked by peptide bonds. The last little thing here is thinking about peptide hydrolysis. So we'll talk about lots of hydrolysis reactions. That's literally the breaking apart by water. So by putting that small molecule water back in, we can break this peptide bond and we can generate free amino acids. So that's going to become biochemistry that's really important as we understand protein metabolism, but we won't study that in detail until Biochem 2. So important chemistry in terms of functional groups reacting that generate our peptide backbone. And then a little bit of side chain chemistry to talk about with the cysteine chemistry that can happen. So again, cysteine being the amino acid that has a thiol functional group. Thiols are the reduced form. So if we do redox chemistry with any oxidizing agent, molecular oxygen here being the simplest, we can have a disulfide exchange reaction that allows for our cysteine residues to become cross-linked with this disulfide bond. Then we have the reduction of whatever our oxidizing species here was, which in this case it was molecular oxygen. So unique chemistry to cysteine as an amino acid, but not unique chemistry to lots of things that we're going to see. And we'll talk about glutathione in a couple of slides here. But just kind of highlighting that redox reactions are very common biologically, particularly for doing some cross-linking. We're going to see thiols in the reduced form being oxidized to their disulfides, and we're going to pair that with the reduction of some species that has the ability to do oxidative chemistry. And so by reducing that oxidizing species, we eliminate its ability to do damage to other materials in our body, like our proteins or our nucleic acids. So just kind of some summary chemistry here between that specific side chain. Not too much I'm going to highlight from this slide. Again, you're not going to have to draw a curve or think of anything like that. We were just kind of remembering here when we saw acid-base chemistry, and we saw what equates to kind of a titration curve, and this halfway point represents the pKa of a molecule, right? The pH that you need to be at for half of your molecules to be deprotonated. So again, the importance of these halfway points, so relating pKa and pH. So when we see something here with amino acids start having at least 2 ionizable groups in our amino group and our carboxylate group. So again, having at least 2 of these functional groups allows us to have at minimum diprotic acids. Many side chains are also going to be ionizable. And then when we start having proteins, we're going to have not only an amino terminus and a carboxy terminus, but lots of side chains that have the ability to be ionized. And so we have the potential to have lots of charge distribution across our protein. So one of the important parameters we're going to think about with proteins is something called PI. And you can sort of loosely think of it as the pKa of your protein. Now again, it's not going to be when necessarily half of your ionizable side chains have been deprotonated, but it's going to be some pH at which the net of the protonation and deprotonation events allows your molecule to have no net charge. And this is really important that we understand there's a difference between a molecule having no charge and having no net charge. We know with amino acids, right? We're never going to see those amino acids that have our NH2 group here and then a COOH group there. That can't happen based on what we know about pKa values and pH. So we might have a situation here where we're going to have species that have no net charge, but they're not necessarily uncharged or have no charge. So big thing is be able to define what PI is and relate it to what it means in terms of the charge on a protein. So again, here's this slide where we talk about some of the chemistry that can happen between small peptides or pieces of protein. So this is just kind of revisiting this disulfide chemistry now in the context of glutathione. So you don't need to know the structure of glutathione. I'd understand the difference between a peptide bond and maybe what an isopeptide bond is. So this is still an amide bond that's made by doing chemistry between a carboxylate group and an amine group, but specifically it is the carboxylate of the side chain of this glutamic acid here. Here's the peptide backbone, right? Our N-C-alpha carbonyl. This is the side chain. It's the R group of glutamic acid, but it has a carboxylate group there. That could do chemistry with the amino group on the cysteine residue, generating an amide bond. And so it's not truly a peptide bond in that it's linking together amino acids in a peptide backbone, but we call it an iso. It's kind of an isomer of a peptide bond that it still generates that amide linkage, but it uses the carboxylate from a side chain rather than from sort of the main backbone atoms that you see for a peptide bond. So I'm not going to go through the details of how that thiol and disulfide chemistry works, but just understand that cysteine in the context of a small glutathione peptide here is a natural antioxidant in your body. So we can have two of these glutathiones that can be then disulfide linked to each other, meaning we're taking some oxidizing species, maybe it's molecular oxygen, reducing that, and so we are eliminating that oxidizing agents or that oxidizing species ability to do damage to other molecules in your body. So again, highlighting down here, this should really say GSH to be glutathione here. But when we have two glutathiones encountering some species that could do some oxidative damage, it sacrifices itself to become disulfide linked. But in order for this process to continue, we need to have some way of regenerating glutathione. And so we're going to talk about what some of those processes are as we move through this course here. And we'll actually talk in more detail when we talk about Tylenol overdose about how that's problematic, but not important for this exam here. Just some other chemistry to think about, not too much in terms of details you need to know necessarily for our in-class exam, but this is just highlighting, and we have some application in lab. When we have certain amino acids here, particularly if we have aromatic amino acids, that allows proteins to have absorbances in the UV region. So that's how we can quantitate our proteins, is we're going to look at the absorbance at 280 nanometers. And that actually comes from the mixture of tyrosines, phenylalanines, and tryptophans, those aromatic amino acids, because this phenyl ring here is going to generate some UV absorbance in that region. Now, what happens here when we have this stretch of amino acids is we have the ability to extend the conjugated system. That makes for a longer, it's redshifted, so a longer wavelength, so we increase lambda max. And we actually push it so far that we get it out of the UV region and into the visible region so we can see things. So again, as we increase conjugation, we're going to take things and allow them to be colored. So lots of molecules that we'll look at that are colored have conjugated systems that are extended, so it pushes that lambda max into the visible region. All right, so that was really all that we had from chapter 4. A couple of things that are important, but I didn't necessarily mention, is when we're talking about chirality in amino acids, that alpha carbon is where that chirality comes from, because we've got 4 different groups attached to it, unless you're glycine and that your R group is a hydrogen. But I do want you to know that naturally occurring amino acids, naturally in terms of occurring in our bodies, are going to be the L configuration. And if you need a little refresher with the differences between R and S for stereochemistry, L and D for stereochemistry, or plus and minus for stereochemistry, remember there's that handout that I provided that can kind of give an overview. Again, not something that I'm going to test you on, but it's a fairly easy read that hopefully can kind of make sure that that's solidified in your brain. Again, we went over this condensation chemistry, thinking about pKa and pi for ionizable groups, talking about that important disulfide biochemistry, and then some kind of applications for extending conjugated systems and for isopeptide bond formation. So moving on into chapter 5, a lot of what we'll talk about in chapter 5 really will save till lab, because this has a lot to do with kind of the sequence of proteins and how we think about that. So a couple of things that are, again, related is a few numbers that I want you to know. So I want you to know the upper and the lower limits for the sizes of protein. Lower limits means you basically have to have enough sequence in order to have structure, because structure affords function. The upper limit, which is 4,000, basically allows us to stay within the limits of error for our machinery. You know, it's hard to string 4,000 amino acids together without making a mistake. And again, solution for this is we create quaternary structure. We allow for more than one protein piece to come together. I do want you to be able to think about how you would do some of this math, right? If you were to have 4 letters that you took one at a time without repeating, the total number of possible sequences is just over 5,000. Know these differences between terms, right? Composition just represents what these letters are, not their order. That implies sequence. And that sequence is what's really important. So sometimes when we talk about proteins, we might just say what the composition is, but we're not specifying the order. So be able to think about these numbers. Be also able to think about these numbers. Now, I don't need you to remember, what the weight of the Earth is or how many atoms there are in the universe, but I do want you to be able to think about how to figure out how many possible options there would be if we tried to string together 100 amino acids or 100 nucleic acids or 100 nucleotides together to make a piece of DNA. So again, how to do this kind of math, 4 to the 1,000. I think those are important sort of just simple math skills to be able to think about. And the big picture thing here is to remind us that nature doesn't even have enough atoms in the universe to test every sequence option to make just 100 nucleotides strung together. And when we think about there being 3 billion nucleotides in every single piece of DNA, nature clearly can't check every option. The last little piece to think about here is just sort of an overview of kind of how we view a dichotomy with protein structure. And that's really on one extreme, thinking of having fibrous proteins, which are long, sort of very alpha helical rich proteins that are just going to play structural roles. And we're going to talk specifically about collagen and keratin. And then proteins that tend to be more globular. So again, these are going to be our catalytic proteins. We're going to talk about them as sort of looking like little globs of bubble gum. Most of the proteins that we're going to study in terms of their function are going to be globular. They're going to be enzymes and so forth. So we will spend a little bit of time talking about fibrous proteins, but then really not revisit that throughout the rest of the course. Most of the proteins are going to be globular enzymes that we're going to study. Again, just a summary here, just glancing through here to see if there's anything I kind of wanted to highlight that I didn't already mention. Again, a lot of this we're going to get to in lab when we do some bioinformatics, thinking about sequences and some structural information. How do we use that information to think about things? So that'll be reserved for lab. So the bulk of what we covered in our protein structure really deals with the 3-dimensional structures. And so that's secondary, tertiary, and quaternary structure. Again, thinking about the stability of protein structure and then how we think about proteins folding. So we spent a good bit of time on this slide. This hierarchy is really important. that you think about primary structure, again, being one-dimensional. Secondary, tertiary, and quaternary structures are all three-dimensional. And again, what differs is what's interacting. And so it's backbone atoms that are interacting via hydrogen bonding to make secondary structural elements. They show an alpha helix here. The two main secondary structural elements that we studied were alpha helices and then beta strands, which interact to form beta sheets. When we have side chains kind of interact with one another, that's how we create our tertiary structure. Hydrogen bonding can be part of that. Remember, that's one of the 4 intermolecular forces that we studied. But all of those intermolecular forces, London dispersion forces, dipole-dipole interactions, hydrogen bonding, and then ionic interactions, all play a role in side chains interacting. And then if those side chains happen to be something like cysteine, we can have disulfide interactions. But again, these are going to be side chains that are interacting within a protein subunit. And the only thing that becomes different when we talk about quaternary structure is it's side chain interactions, but now between protein subunits. So again, this is only going to be occurring if we have a multi-subunit protein, but basically the same interactions are possible for either tertiary or quaternary structure. So I didn't kind of highlight this. So what holds kind of primary structure together? Primary structure is akin to saying the sequence of a protein, and that's the peptide bond. That's a covalent bond. And when we talk about primary structure, we're really talking about sequence of amino acids, and that, again, we talked about in chapter 5. Ramachandran plots. So you're not going to need to know how to draw a Ramachandran plot. Basically, I just want you to understand that when we have this protein backbone, that we're hanging side chains off of it, and we're trying to think about what are possible structures, 2-dimensional or 3-dimensional structures that we could have for these proteins, is that structure formation is going to be driven by 2 things. Sterics, we don't want things to be able to bang into one another, and that's what a Ramachandran plot is really about. It's about allowed sequence or allowed space that avoids steric clashing of atoms. So again, if we imagine making an alpha helix out of this backbone, right, there's going to be certain allowed angles between these atoms along the backbone that are going to ensure that we don't have side chains and backbone atoms sort of banging into one another. And if we form a right-handed alpha helix, we're going to live in this sort of allowed space. If you twist your helix the other way, you're going to live over here. And you do not need to know, the anatomy of a Ramachandran plot, but I want you to know that it basically looks at 2 sets of angles and shows what is sterically allowed. So again, when we talk about structure formation and the part of it that's steric allowance, a Ramachandran plot just shows us where these allowed islands are. Again, looking at beta sheets, it's going to be a completely different structure of your backbone, because remember, for alpha helices, we called those coils. For beta sheets made from beta strands, we called those zigzags. And so to make a zigzag out of a backbone is going to be a very different set of angles than if you're making a coil. And so we have these allowed sort of islands of sequence or of space that allows structures that won't have things banging into one another. So again, that's half of structure formation is making sure we don't have stuff banging into one another. The other part of it is making sure that if we are freezing out kind of molecules moving more. Remember, entropy is more molecules moving more, and that's going to be unfavorable. That's going to be unfavorable energetics. So at the end of the day, if we want to make structure, we have to optimize energetics, and that means we have to think about both entropy and enthalpy. Entropy for structure formation is always going to be unfavorable because we're freezing out molecular motion. So we're not going to have more molecules moving more. They're actually going to be moving less. So we must have some type of enthalpic compensation. And remember, for enthalpy, we talked about bonds and interactions. So for this process to occur, if we have unfavorable entropy, we must have favorable enthalpy. And so we call this entropy-enthalpy compensation, but kind of flipping back here, this is what all of these are about. Hydrogen bonding to hold helices and beta sheets together. All kinds of intermolecular forces or even covalent bonds to hold together tertiary and quaternary structures. If we want to make structure, we want to have order in our system, that's energetically unfavorable. We need to compensate for it by favorable enthalpy. So again, now elaborating a little bit more on specific secondary structural elements. So this slide allowed us to contrast what we see structurally for alpha helices. So we show both of these as a ball and stick model as well as a space filling model. Some commonalities between what we see for both the alpha helices and the beta strands is that they're held together by hydrogen bonding. And that hydrogen bonding, again, is between backbone atoms. So it's between the carbonyl of a backbone and then the amide nitrogen's hydrogen. So again, backbone atoms, we can see that that hydrogen bonding is going to be intrastrand or within an alpha helical strand. And then for our beta sheets here over here, remember some terminology here. Beta strands are the zigzags that we have. At least 2 strands interact to make a beta sheet, which reminds us that that hydrogen bonding is interstrand, within strands. And so we can see that hydrogen bonding that we have right here. And the strand that we've shown here is anti-parallel. We can see that this one is going N-C-alpha carbonyl, N-C-alpha carbonyl, right? If we've got N-C-alpha carbonyl, N-C-alpha carbonyl, N-C-alpha carbonyl. This one goes this direction. If we do the same thing, we can see this other one goes in the other direction. So this is an anti-parallel beta sheet. We're going to do a lot more with structure in lab when we have our bioinformatics lab and kind of look at structure. But again, contrasting here, both hydrogen bonding that we see for alpha helices and beta strands, but it's intrastrand for alpha helices, interstrand for beta sheets. What's common, and again, this should have probably been a commonality between these, is we're going to have regular, repeating, and allowed phi and psi angles. So remember, those phi and psi angles are the angles that we have on our Ramachandran plot. So basically we're saying we need to have sterically allowed, but in order to have like a regular structure like we see when we have a coil or we see a zigzag here, we have to have the same bond angles. We have to have a pattern, a repeating pattern. So a regular repeating pattern that's sterically allowed will give us an alpha helix when we have a certain shape. And different regular repeating and allowed phi and psi angles will give us the zigzags, which allow us to make beta strands that interact for beta sheets. So again, when we look at these structures, we want to think about zigzags being our beta sheets and coils being our alpha helices. So one of the things too that is, I think, part of your homework is the idea of when we look at these in terms of ribbons diagrams, we're going to see coils still for our alpha helices, but we're going to see arrows for our beta strands there. So just ways that we need to make sure we know that we're looking at a specific structural element in proteins. So just another sheet sort of comparing what we see for alpha helices and then beta sheets when we're talking about where side chains are. So these show us that we have kind of directionality to where we might see side chains. So side chains for alpha helices point out and down. This is just a reminder that even though we look at this structure and it looks like there's a giant hole in the center of this alpha helix, really there's not. If we look at the space filling model, we can see that these gold atoms all represent side chains. They're all pointing to the outside of the alpha helix. And we can kind of see that again here. They're pointing outside and they point out and down. The side chains for a beta sheet or a beta strand, we can maybe see it a little bit better here. They alternate on opposite sides of our zigzag. And so these are on alternate faces of our beta sheet. And what that allows us to do is sort of have a topology or a geography, if you will, of where we're going to have our residues. So we have, we can have a sidedness to our alpha helix or our beta sheet here. And again, commonality is that we're going to see all of our side chains, whether they're on alpha helices or beta sheets. We're going to represent them kind of as dangles on a backbone, and where they're going to live depends a lot on their hydrophobicity or hydrophilicity, if you will. So things that are hydrophilic, water-loving, like to be on the outside. Things that are hydrophobic like to be on the inside. That is the premise of the hydrophobic effect, right? Hydrophobic effect is defined as the tendency of nonpolar substances to minimize their contact with water. So again, if we're going to minimize our contact with water, we're going to put those side chains on the inside of a protein. So all of these red amino acids represent hydrophobic amino acids. We can see that they're largely on the inside of this protein. Green, which are hydrophilic, are on the outside. So you are going to see questions on your exam that kind of ask you which protein is more likely to be found on the outside or the inside of a protein. There are problems like that on your homework. You're going to be asked these by single letter code. So again, it's going to be important that by the time you take your exam, you're pretty proficient with knowing your amino acid side chains by their single letter code so that you'll be able to answer those questions. Moving now into just a couple of the specific proteins that we studied in this chapter. Again, we're going to focus on the two fibrous proteins, keratin and collagen. We'll have lots of time to look at different globular proteins, which tend to be enzymes, in the rest of the course. So when we start with alpha-keratin, here's the things that you need to know for, again, Mondays or the exam, whenever the exam is going to happen to be. Keratin is very cysteine rich. Okay, so that allows us to do some cross-linking chemistry. And we talked about that in the context of hair chemistry. You don't really need to know much about that for the exam. It was just kind of a neat example to think about kind of sulfur chemistry with regards to hair. But here's some of the important things that you need to know about keratin. Keratin is two right-handed alpha helices in a left-handed coiled coil. Okay, so we can see the structure of this here. They're two right-handed alpha helices. That's the kind of more prototypical alpha helix. And then they're going to wrap around one another in a left-handed coiled coil. We're highlighting this here because remember we see this theme that nature knows that if you want to make strong sort of fiber strands, you're going to have more than one strand that you wrap in opposite directions. So another kind of important piece to think about here is when we have alpha helices, and we start, for example, at position 1. This is position 2 of an alpha helix. Position 3, keep in mind we're looking down the axis. So if you were to have a cylinder that's an alpha helix and you were to kind of look down the center of it, remember our side chains are going to be pointing out here. So this is the center of our alpha helix here. Side chains are pointing out all along here. So this is the first position, second position, third position, fourth position, 5th, 6th, 7th, and once you get back to the 8th position, you are superimposed back on your starting position. So what this shows us is that the first and the fourth position can lie on the same side or face of our alpha helix. So we might see, for example, if we wanted to create a greasy or a non-polar face to our alpha helix, that we'll put the same residues in the first and the fourth position. And that's exactly what keratin does. And so that creates sort of a greasy strip, if you will, on one side of our alpha helix. And it's going to find a greasy strip on another one of these right-handed alpha helices. And so that facilitates the making of this left-handed coiled coil by, again, these non-polar residues wanting to minimize their contact with water, and so they will find and associate with one another. And that creates a little hydrophobic center to this left-handed coiled coil. So some structural details there for keratin. Again, cysteine-rich protein. That's an important piece to remember. Again, less all the application details that we talked about in terms of how you perm hair and all of that, but I do want you to remember that keratin is cysteine rich. We talked a bit about collagen. So collagen, again, if we think about this whole idea of wrapping things in opposite-handedness, we take that left-handed alpha helix. That's going to be a tighter alpha helix, going to have some specific things that we need to think about in terms of what's going to be allowed in that tighter alpha helix. But we're going to have 3 of those left-handed helices that we wrap in a right-handed triple helix. So we can sort of see this over here in a very kind of artistic rendition of this. So each of these represents a left-handed alpha helix, and it's wrapped in a 3-handed or a right-handed triple helix, so 3 of those strands. So one of the things that's unique in terms of the makeup of collagen is the 3 amino acids that we tend to see there. And we tend to see them in this repeat. Glycine, XY, glycine, XY, glycine, XY. Glycine we have there because it's the smallest amino acid. Remember I told you for these left-handed alpha helices, we tend to have some steric constraints. So there's only going to be some very specific amino acids that can fit there. Glycine fits there because it's small. Proline fits there because remember, proline has that unique backbone that has the side chain attached to the backbone. That allows us to have a cis geometry. We didn't talk too much about geometry, but most amino acids tend to have what we call trans geometry, which puts the back or the side chain sort of opposite from one another. This cis geometry is facilitated by proline, but that's why you don't find proline in a lot of different amino acids. What you're going to see here is proline is very prevalent in collagen because it allows us to have these left-handed alpha helices. And then we also tend to see hydroxyproline or hydroxylysine. So again, some more proline to allow for that unique sort of structural feature there with our left-handed alpha helices. And then we see some hydroxylysine. So again, here's a hydroxylysine. What I'm going to just kind of mention very briefly because it comes up later when we talk about one of our reaction mechanisms that you haven't yet gotten to. This lysine residue here has the ability to undergo some chemistry via enzymes that will convert this amino group into an aldehyde. And when you have this long chain here with an aldehyde on the end on one of these chains, and then you have an adjacent chain that has another one that's undergone the same chemistry, you can actually cross-link collagen fibers via this aldehyde residue. You can actually do an aldol condensation, and so collagen can be cross-linked. And as animals age and tissues age, your collagen gets more and more cross-linked, and that results in sort of tougher and tougher tissue. So that's sort of what happens with very old meat in old animals, as it gets tough compared to sort of a younger meat. So again, this is a very abundant protein in skin. You'll obviously hear about a lot with collagen and women. Part of one of your case or your take-home questions is to be thinking about collagen supplementation. But little side story that we talked about with collagen is to make these two residues and make these hydroxylated versions, you need vitamin C to do so. So we talked about vitamin C deficiency resulting in scurvy. And so again, not having enough vitamin C means you can't make enough of these residues, means you don't make good collagen, and again, you don't have then good connective tissue in gums, in your skin, and so forth. And so we talked about sort of the disease implications with that. We also sort of talked about some collagen diseases. People who are hyperflexible or have elastic skin tend to have collagen deficiency diseases where they don't make sufficient collagen, or we talked about the disease osteogenesis imperfecta, which is really brittle bone disease, which means you don't make sufficient collagen in your bones are very brittle. So just some neat applications of collagen there. Again, we didn't talk about collagen supplementation. We indicated we were going to save that for a couple of questions that you'll see in your take-home. And then we had our insulin structure case study. So a lot of what we talked about with our insulin structure are things that we sort of highlighted with a lot of things here. I do want you to know what the structure of insulin is, right? We start out with proinsulin, which is a single chain. We do some enzymatic work by peptidases that are in your beta cells, which are in your pancreas, to liberate this free sort of C-peptide and then functional insulin. So I do want you to know what the structure of insulin is, a little bit about if you were to look at different amino acids that we substitute in, why certain amino acids might affect the PI of a protein, why certain amino acids might be more conservative substitutions. So be able to think about the amino acid functional groups. Again, less important that you're able to recognize and think about every single amino acid structure, but importantly, know their functional groups. So if you were to see something that were to ask whether this would be a conservative substitution, like we see, for example, with serine and threonine, or something that's likely to have a significant change, for example, introducing a charged amino acid like we see here. So thinking a little bit about how this relates to gels. Again, this is really more related to things that we'll see in lab. But let's see, just kind of flipping through here and seeing if there's other things that I want you to be able to think about. Thinking about the difference between something being folded and something being functional, right? And the concept of a native fold means that it's folded into its functional state, right? We can have something that's folded, but again, if it's not in its active native state, we're not going to be able to do functional biochemistry with that. So again, the importance of having the C-peptide in insulin, in this case looking at RNase A, sort of the right pairings together with these disulfide links. So again, the purpose of these disulfide links is to impart specificity. It does not contribute to stability in proteins, it's specificity. Let's see, you're not going to see anything on immune system on your test. And we talked about sort of what the purpose of the C chain is. And I think everybody got that by the end of the case study, that the purpose of the C chain really holds the right cysteine residues together so that we have the right specificity. And those disulfide links aren't important for stabilizing the protein, but they are really important for dictating who is next to who. And again, we talked about structure determination. Just very generally know the differences between X-ray crystallography and NMR, which is the one that's a frozen image, which one is a dynamic image, and sort of the pros and cons of them. You had a homework question sort of thinking about that. That X-ray has very high resolution structures. So again, high resolution means a low number here. So we know them with highest degree of accuracy, but again, you need to be able to crystallize something, and there's some criticisms that, is this really what this molecule looks like in solution if you've made it sit still? NMR is great because you don't need to crystallize something, but you can't have very big things. So we'll get into a lot more detail with this when we do our bioinformatics and structure determination lab, but just very generally the differences between X-ray and NMR for this test. We've sort of talked about this already, sort of revisiting this, what holds proteins together. What really contributes to the stability of protein structure is the hydrophobic effect, so putting those greasy residues on the inside. So again, this slide was a great one to summarize the differences between stability and specificity. So stability is, again, what is the enthalpy that really holds structures together? It's the hydrophobic effect. So that's going to be our London dispersion forces. That's what really contributes to stability. Specificity is really who is connected to who, who's next to who. And we can have all of these other guys that sort of contribute to that. You know, specifically who is next to who when you make the primary structure of an amino acid. That's a peptide bond that holds that together. But we also can have disulfide links that sort of hold things together. Salt bridges really are an ionic interaction in proteins, and we've got lots of hydrogen bonding that can either hold secondary structures together, but we also see that in tertiary and quaternary structures. Just a little bit more, talking about this 100 mer. We don't have the time to test every single possible option. Nature is efficient, and so it doesn't really walk randomly. It has specific sort of motifs that it's going to sort of work to fold, and kind of it's going to have variations on a theme. It's not going to have a lot of different ways that it does things. But we do want to think about kind of falling into these traps of something that's folded but not functional. And we can kind of get ourselves trapped into these folded but not functional areas. And we'll talk a lot about this in terms of Alzheimer's disease and so forth. Native, meaning again folded and functional. So not a lot to really think about with gluten for the exam. I want you to remember that gluten really represents two proteins that are disulfide linked together. We have a protein, or there's a protein here that we cannot digest. So normally kind of digestive function is to take proteins in this case, denature them in your stomach. Your small intestine has lots of peptidases that get them down into individual amino acids. That's important because if the only way to get something out of your GI tract is to convert it into a monomer and get it through channels and gates, if you will, that are in that one cell thick, that is your intestinal cell wall. But again, if you've got holes in that one cell thick intestinal cell wall, you can have larger pieces get through. That can elicit an immune response, and that's very problematic. So just very superficial stuff with gluten, just kind of what it is and the importance of degrading things to monomers in order to get them out of your GI tract. Much more on this in your take home. Same thing with diabetes. Again, just a little bit about diabetes. Diabetes really is the formation of these insoluble plaques, right? This is a response to sort of an acute insult in your body, and this serves an important function. It sort of downsizes the functional space in your brain. We talked about kind of ways that could happen, but this is meant to be short term. Your body will then clear these plaques out and get back to sort of full functional space. The problem is if these insults are chronic. The problem is if you have a genotype, you have a SNP that is not conducive to those clearing, if you're not getting sufficient sleep. all of these sort of impact your ability to clear what is meant to be a short-term response or to an acute, a short-term insult. So again, very superficially, if anything, will you see gluten or Alzheimer's on your in-class portion, high focus though on your take-home portion. And that is it then for what we're going to see for our third learning unit on protein structure.